get in here. And we'll get started. Okay, it looks like you might get a few more coming in over the next couple of minutes. Actually, I do see some more people entering, but we're going to get started. Thank you for joining us on this beautiful fall morning. My name is Jessie Jones, and I am the coordinator for the Coast Watch program of Oregon Shores Conservation Coalition. And I am very happy today to talk to the newly formed PRIMED network. And we have a number of panelists joining us today. They're going to share some really interesting and great information about marine diseases. I know that I'm excited about this one, and I know that a lot of you that have contacted me for a link are also excited. Remember that 1 week from today, same time, we will be having another one of these, but more of a training webinar, and we'll be talking about that too. Oregon Shores is a regional group that works to protect the environment of the entire coast of Oregon, from the Columbia River all the way down to Gold Beach, and from the tip of the Coast Range out until the edge of the continental shelf. In addition to organizing Coast Watch and citizen science, Oregon Shores tackles a wide range of conservation concerns, including land use planning, water quality, endangered species, shoreline management, and marine conservation. Through Oregon Shores Coast Watch program, volunteers adopt 1 mile of the Oregon shoreline, and they keep watch for a wide range of natural phenomena and human impacts. Coast Watch also organizes a number of citizen science projects. And new volunteers are always needed, so you can contact me. Let me know, send me a chat or an email after this is done if you are not currently a Coast Watcher, and I will be happy to share with you more about our program. So at this point, I am going to introduce Dr. Sarah Gravem, who is the founder of PRIMED, and her team, and they are going to take it away from here. So I'm going to do a quick stop share. There we go. Oh, and Tim made it. Welcome, Tim.
Oh, thanks.
Okay, and then I think Sarah will be introducing folks, or maybe Laura will, and I am going to stop my video at this point. All right.
Hi everyone, my name is Sarah Gravem. I am a researcher at Oregon State University. I've been there about 5 years, and I am the founder of the PRIMED network. So PRIMED means Primary Responders in Marine Emergent Disease. We're going to be talking to you a bunch about our network in the second half of today. The first half of today will be kind of a crash course on marine diseases. And I want to introduce first the panelists here. So I'm Sarah Gravem, like I said. I have been in Oregon as a researcher at OSU for 5 years, and I came here from doing my PhD at Bodega Marine Lab, which is a UC Davis Marine Lab. I did my master's at Cal Poly San Luis Obispo. I did my undergrad at UC Santa Barbara, and I grew up in the Bay Area, East Bay in Concord, and I spent a lot of time as a child accompanying my dad on abalone diving trips and being left in the tide pools to entertain myself. And so that's where I fell in love with the ocean. I also grew up in a hunting family, and so I have a connection to terrestrial nearshore environment, and I love the way that the ocean provides for us and the way that we can help maintain the ocean. And so with that, I'll introduce my co-panelists. First up is, I think, Laurel Field.
I wasn't sure how to unmute myself. Okay. So hi everyone, my name's Laurel, and I was born and raised in Oregon, actually in Corvallis. I left for a bit, went to college in Washington, And I feel so lucky to have ended up back here in the areas I grew up loving and continue to visit today. I recently started a master's program at Oregon State in marine resource management, and I will be studying effectiveness of marine protected areas and management of those areas. I'm also a faculty research assistant at Oregon State in the Mengi Lab, and I study effects of sea star wasting on intertidal communities in both Oregon and Northern California. I took this job like right out of college and moved back to Corvallis. And so it's my third and final year of this position, but I love it so much. I get to spend so many days of the year out at the coast doing intertidal research, and I actually go on my next trip tomorrow, so that's very exciting for me. And lastly, I'm a PRIMED steering committee member, and definitely the disease ecology work I've gotten to do at Oregon State has inspired my interest in marine disease and also allowed me to join the PRIMED network, which has been great. So thank you. And next, I think, is Silke.
Hi, everybody. My name is Silke Bachhuber. I use she, her pronouns, and I am a grad student at OSU in the Mengi-Lubchenco lab with all of my esteemed panelists today. I'm a member of the PRIMED steering committee, but my broader research interests include community ecology, climate change, and the impact of climate and disease events on the structure and function of our ecological communities and how they'll look in the future. I grew up tide pooling with my dog on the Washington coast and developed a real passion and enthusiasm for marine biology, and especially for our beautiful and diverse rocky tide pool ecosystems, and then moved on to do my undergraduate work at the University of California, Santa Barbara, just like Sarah, although we didn't quite overlap. And then moved back to Oregon to pursue my PhD, actually just almost exactly 4 years ago. And throughout that time, I've been lucky enough to work in some very cool places and with some very cool organisms, including, oh, sorry, Jesse, let me turn my video off too. Including sea stars, abalone, urchins, limpets, and I also, as part of my dissertation work in New Zealand on a very special little snail down there. And I think that's all I have.
Hey everyone, my name is Lily Mikesel, and I'm an undergraduate senior at OSU studying biology with an option in ecology. I'm a huge animal lover, and that's what really sparked my interest in biology. I have two really sweet dogs named Lizzie and Envy. They were my first pets, and wanting to learn more about their behavior and emotional intelligence made me curious about other creatures on this planet. I was born in California, I grew up in Illinois, and I moved to Oregon 4 years ago. I've always felt drawn to the beach and love being around water, but my interest in marine biology and ecology really grew when I moved to Oregon and just started exploring the beautiful coast. And I think next is Bill.
Hello, this is William Baxter. I guess when asked to describe myself, I like to use the word curious. I'm curious about the world and all of its wonderful places and inhabitants. So I was once introduced at a webinar as a jack of many trades and a master of few. So based upon that, I enjoy traveling, photographing, and exploring this planet. All of these are just futile attempts to satisfy my curiosity of this amazing world. And coming into this, also being married to a fellow scientist, it increases my knowledge of marine life, which I love. And I'm very happy to be part of this project, and I'm very happy to help them with their data. And I think that I'm the last one, and thank you. Oh, excuse me, Tim.
No worries. Hi, everyone. I'm Tim Miller-Morgan. I'm an aquatic veterinarian with Oregon Sea Grant and the Carlson College of Veterinary Medicine. I've actually been really fortunate to be involved in sort of with aquatic animals from a husbandry and wildlife and veterinary standpoint for actually about 30 years now. I was real fortunate to be able to start out in high school working with at Point Defiance Zoo and Aquarium, and that just sort of spurred my interest. And I've really been fortunate enough to work actually all over the world with a variety of aquatic animals, but I've always really been interested in kind of wild populations and assessing their health and figuring out ways to manage their health. Pretty much been worked in aquaculture, wild fisheries, captive environments, public aquariums. I had a little detour for a while where I worked with grizzly bears and black bears, but then came back to aquatics. So I've been able to work on everything from killer whales to abalones, and it's been really exciting. I'm always, I guess, I would kind of agree with Bill. I'm pretty curious and always interested in what can we learn more about these animals, particularly in terms of managing their health. So I'm one of the advisors for the PRIMED network, and if we can switch to the next slide, I can introduce the other advisors too. So a couple of my colleagues that are also advisors, Dr. Christiana Lohr. She's a professor at Oregon State University, also at the College of Veterinary Medicine. And then Dr. Stephen Rumrell, who's the shellfish program leader for Oregon Department of Fish and Wildlife.
Okay, hopefully everyone can hear me. This is Laurel again. So I'm going to lead us in a couple of rounds of disease trivia to start us off today before we get into more in depth about marine disease and primed. So this is just for fun and to get us thinking about disease, so no worries if whether you get it right or not. We'll be using a Zoom poll. So once I explain the questions, there's only 4. You'll be asked to select your answer, and then we'll be able to show percentages of what everyone thought was the answer. And so, and then I'll explain the correct choice. So we will get started, and hopefully this works as we planned. Okay, so the first question is, which sea star is exhibiting symptoms of sea star wasting syndrome? And so you can choose options A through C are these individual stars, or if you think all 3 are wasting, option D is all of the above. And for anyone who's not familiar with sea star wasting, it's a disease that causes deterioration of the tissues of the sea star. So we will launch our first poll now, hopefully. Great. If anyone doesn't see the poll that just popped up, please let us know in the chat. And we will give everyone about 30 seconds to select their answer, and then we'll check it out after that. So I'll let you know when we're close to being done.
It looks like, this is Jessie, it looks like folks have voted, so I'm going to share the poll.
Great. All right, so most people said D, all of the above. Okay, now I'm gonna switch slides. All right, so the answer was a little tricky. The answer was A. And so A we know is wasting because we can see those white lesions on most of the arms of the sea star. B was a trick because that sea star is actually spawning. And you can see, first of all, it's not quite the same lesion, but there is that substance, and that's like more orangish. And also it's a little tricky because you can see that black sort of looking tissue on top, but that's actually algae on the sea star, so it's still healthy. And then C was also sort of a trick because we know that losing arms can be a symptom of wasting, but in this photo it's hard to tell. It could have been lost through another way. So yeah, so it's D, all the sorry, A, just A is wasting. All right, so that was the first one. So we're moving on to question 2. Thanks everyone for playing along here. So how many viruses can there be in a single drop of seawater? A, 10, B, 1,000, C, 10,000, or D, 10 million? So we'll relaunch the poll. Okay, there we go. And Jessie, I'll just let you close it when it looks like people have answered because I can't see.
Okay, great,
Yeah.
Will do. Got the answers coming in. These are fun.
Yeah. All right, great. So it looks like most people said 10 million and some people said 10,000. So congratulations to everyone who said D. it's 10 million. This might not be on average, but there can be up to 10 million viruses in a single drop of seawater, which is pretty wild. Okay, that was question 2. So now, okay, our third question, and we'll have one more after this, is which is responsible for toxoplasmosis disease in sea otters? And Toxoplasma gondii is a protozoan parasite that can cause fatal brain infections in otters. And it can also cause similar outcomes in different animals, but for now, with this example, we'll just focus on otters. So our options are A, cats, B, turban snails, which are a marine snail, and you can sort of see it has a more pointed shell. C is giant kelp, and D, you think all 3 of these organisms may be responsible. So we'll go ahead and launch this poll. Okay, it looks like everyone answered. Okay, so most people said B, which is the snail. Great guess. And then there's also split between A, C, and D. So those who said D were correct. So this is just one mechanism by which otters can become infected with toxo. There are other mechanisms, but in this example, and this has been studied, there's actually a relationship between cats, kelp, snails, and then finally the unlucky otter. So cats are the hosts of this toxo, and their toxo can enter the marine system through feces. And once in the marine environment, egg-like cysts that are of toxo actually stick to the mucus layer on kelp. And then as snails graze that kelp and consume the mucus layer, toxo is transferred into the snails. And while the snails aren't infected in the same way by the toxo, an otter that comes along and eats that infected snail will then can be infected with toxo itself. So congrats to whoever knew that or guessed that. That's, yeah, it's a cool example. Okay, so moving on to our final question is which is responsible for bleaching in the coral Oculina patagonica? And quick disclaimer, this coral is not from this region. This is from the Caribbean. And so our options with this one are the bearded fireworm, A. B is Vibrio chaloi, C is parrotfish, and D is, you think it's both the fireworm and the bacteria, the Vibrio. Okay, so we'll launch our final poll. I wish we had that quiz music in the background next time.
We'll make sure to do that. We'll work on maybe some ocean music,
Yeah.
maybe some whale songs.
Mm-hmm. Something soothing. All right. Okay, so it looks almost it's tied between B, the bacteria, and D, both A and B. So it seems like some of you caught on to my tricks here because the answer is both A and B. And so the way this disease scenario works is that it is the Vibrio, the bacteria, that causes the bleaching in the coral. And the way this works is that Vibrio inhibits the photosynthesis of the symbiotic dinoflagellates that live in the coral and are essential to its health and survival. But what happens is that the bacteria cannot survive in the coral as the temperatures drop below 20 degrees C. So in winter, this bacteria will actually live in the fireworm and survive through the winter. It won't infect the host in the same way because there's no symbiotes. But as the temperatures warm again in summer, the bacteria will then reinfect the coral and cause the same bleaching phenomenon. Okay, so Congratulations to whoever got that last question. And thank you for playing, everyone. I hope we learned something and also had fun. Marine diseases are truly diverse as they are complex. And to get into more of those complexities, I'm going to turn it over to Silke, who's going to talk a bit about disease ecology.
Awesome. Thanks, Laurel. So you've learned a little bit about some really fascinating examples of the different ways that marine diseases can impact our really valuable marine ecosystems. But I want to kind of take a little bit of a step back and ask more broadly why we care so much about diseases in the ocean. And there's a number of reasons for this, and some of them are similar to why we care about diseases of both people and animals on land, and some of them are a little bit unique. The first one, and probably one of the most familiar, is that marine diseases directly affect commercially valuable species. And some of the more high-profile ones that you might have heard of include salmon, especially salmon in high-density aquaculture systems, and shellfish, but they can also have cascading effects that impact the rest of the ecosystem. And I'm going to talk more about this in just a minute. Marine diseases also impact the structure of ecosystems, which is a term that ecologists often use to describe essentially just who lives where, as well as the services that ecosystems provide to humans, which is an umbrella term that we use to describe ecosystem benefits. And that includes in Oregon, things like food, recreational opportunities, and actually shoreline armoring is a really important one in many of our coastal communities as well. And then we also care as researchers a ton about marine disease because we expect to see an increase in both the frequency and the severity of these disease outbreaks over time. And as climate impacts intensify, we're seeing these increases both on land and in the ocean. And again, as I mentioned, they can have these cascading effects. So to set up kind of the example that I'm about to discuss in more detail, I've included this picture on the right of the trophic levels or the feeding levels that are present in many of our ecosystems and that you might already be familiar with. So if you've seen Lion King, the original, or the live action remake recently, you might be familiar with some of these animals. We have carnivores like lions, herbivores, which consume plant matter, and that in this example would be zebras and gazelles. And then finally, we have plants, or our primary producers that are a source of energy from the sun. And in the ocean, we have a very similar, again, this is really highly simplified, but we have a really similar setup in terms of the structure of our communities. And Laurel, will you hit the next slide for me, please? Thank you. With just some slightly different species and some slightly different organisms. And so many of you might be familiar with sea star wasting syndrome in our coastal habitats, and you might know about their effects on our really iconic intertidal predator, the ochre star or piesaster. Some of you might already be aware of the interaction between these ochre stars and their mussel prey as another common example of where disease has impacted trophic interactions, so interactions between these various levels in our trophic hierarchy. And I'd like to share another story with you about a slightly different sea star, the sunflower sea star, which we also refer to as Pycnopodia. Like the ochre sea stars, it was hit really hard by sea star wasting in 2013 and 2014. And can I get that next slide, Laurel? Sweet. So these sunflower stars were strongly impacted by sea star wasting. And in our subtidal habitats, they're almost a little bit like lions in that they're very, very, very voracious and actually very fast-moving predators in our subtidal ecosystems. And they're not consumed by humans, but they are really important and they're involved in a trophic cascade that I'm about to describe in a little bit more detail. So again, we saw this huge population decline in sunflower stars as a result of sea star wasting. And there's actually some evidence. Sarah's working on another project where there's the potential that these sea stars may actually be listed as endangered species because of how severely they were impacted by this disease. So we saw huge population declines in the sunflower stars. And again, we don't eat the sunflower stars. They're not very appetizing, but they do affect species that we really care about a lot as a result of this trophic cascade. So we have our predators consuming our herbivores, which in this case are sea urchins, purple sea urchins, and they in turn consume algae, including our really beautiful and iconic kelp forest kelp nereocystis, which you may have seen either out kayaking or snorkeling or fishing. or just washed up onto our beaches as wrack or drift algae. Next. And so when we saw this decline in predators, we saw corresponding and cascading impacts on our ecosystem, where when there's less Pycnopodia, fewer sunflower stars out eating sea urchins and actually also scaring them into grazing and eating less algae. Next. We see these huge booming increases in sea urchins, and that corresponds then to a decline in their primary food source, which is kelp. Next. And oh, that's an extra animation. And so this can create a phenomenon known as an urchin barren, where urchins completely overtake kelp forest ecosystems and cause significant declines in kelp cover. As you can see here on this image on the right, this sort of tide of purple urchins is coming in. And the result of these urchin barrens is that you often will end up with communities that they go from these very lush, very vertically distributed kelp forests all the way down to essentially bare rock, which is the little bit of algae that the urchins sort of come across and scrape almost as soon as it grows. And so a combination of these urchin barrens and warming waters threaten kelp. And it's important to note that these urchin barrens are part of a natural process in our ecosystems, but we're seeing more and more of them as a result of this disease event, as well as climate change. And instead of balance in our trophic structure, we're seeing this takeover of urchins, and they're eating all of our kelp. And we care about this, and this is really important because kelp forests are unique, diverse habitats that have their own ecosystem structure. They're incredibly productive, they're highly diverse. Many of the fish that we love to eat spend all or part of their lives in a kelp forest, and they provide habitat, food, and shelter for rockfish, crabs, birds, marine mammals, and hundreds of other species. Kelp also provide many ecosystem services to humans, including sheltering our coastal habitats from large waves and storm events that we see on the winter along the coast here. They provide food for us and the things we like to eat. And they can actually even buffer the impact of ocean acidification. And finally, sort of in the context of your beach walks, the way that you might see kelp the most often is that finally kelp wrack is a really important nutrient source for our sandy beach and dune ecosystems here on the Oregon coast. So I don't think I have to spend too much more time emphasizing how important kelp is to all of you, but I wanted to share this example as a way that the effect of disease on a top predator that we don't eat has significantly affected the ecosystem services and the structure of our coastal habitats, with significant and long-lasting ramifications for our coastal habitats and the benefits that we derive from them. And just stepping away, go ahead and to the next one, Laurel, from our ecology example for a moment, I wanted to just briefly highlight an example of community science and collaboration that's come out of this sort of change and emergency that we've been sort of detecting and observing, where we have this significant decline in our kelp forest habitats. And that includes the Oregon Kelp Alliance, or ORCA, which represents diverse interests in our kelp forest ecosystems with the end game of supporting Oregon kelp forests. And in California, interdisciplinary groups are working together to preserve and protect our kelp forest habitats, including the recovery plan I've included on the right, which is the result of collaboration between actually our National Marine Reserve System and the California Department of Fish and Wildlife. So lots of opportunities for some really exciting interdisciplinary and collaborative work in this realm. But it's important to remember that even these sort of small perturbations in ecosystems can have these cascading trophic effects that have significant ramifications for our coastal habitats that we all know and love. And with that, I'll hand it over to learn a little bit more about the specifics of marine disease. Thanks, everyone.
Okay, thanks everyone. This is Tim Miller Morgan, and Silke gave an overview of kind of your disease ecology and some really nice examples. And one of the things that we're seeing now are an increase in marine diseases, and there are a number of factors that seem to be associated with this. One being climate change. We're seeing temperature shifts. Increased temperature certainly can compromise some of these animals, suppress their immune system. We're also seeing hypoxic events associated with this, and again, compromising and stressing animals, so that will predispose them to diseases that are out there. And certainly pollution is an issue. It can outright affect animals outright, or again, it can suppress them and make them more susceptible to diseases. The other thing to consider is, we oftentimes move these animals around, where we may collect them, we may move them to other places. They may get released either legally or illegally and may move diseases around that way, introduce diseases to new areas. So these are all factors that have some potential role to play in our sort of increased observation of marine diseases right now. But studying marine diseases or identifying marine diseases can be somewhat challenging. And let's talk about some of those reasons next. So typically when we think about diseases, usually we're thinking about terrestrial animals, animals that are living in the air. When you put animal populations in the water, first of all, they're a lot more difficult to catch. They're a lot more difficult to see. And then disease agents can actually spread much more quickly and easily in the aquatic environment. They can move in currents, they can have storms, they can move around that way. The other thing we'll often see are animals, they'll gather in large numbers in the wild, or if they're cultured. Sarah mentioned, aquaculture earlier. We're typically putting animals together into large numbers in confined spaces, and that can certainly promote increased disease movement between animals. But also we think about some of the invertebrates that are sessile, they're basically, you'll have populations or colonies, and disease can move quickly around those populations. They can also concentrate the diseases in one particular area. So these are all things we also need to think about. These animals, when they're sick, are difficult to isolate. A standard approach, if we're dealing with a captive population of animals on land, is we remove and quarantine the sick animals. It's very difficult to do in a wild situation. The other issue is that it's often really difficult to detect these diseases and then to characterize them. We just don't know a lot about these diseases, and we're learning, but the amount of information we have on the common diseases of aquatic animals is much less than it is for terrestrial animals. And I think one of the other issues too is we just don't have baseline information on a lot of these diseases, as well as on many of these animal species. Next. I alluded to this already. We have also many of our animals, particularly invertebrates, but some of the vertebrates have, traveling, back one, thank you. They have millions of larvae that move around. Typically there's low survival, but because these animals are moving around in the aquatic environment, riding on currents and that kind of thing, they can spread the disease as well. And oftentimes what can happen when you have a disease introduced into a new area is you can have extirpation, which is basically a localized extinction of a particular population, or you can have a more broad extinction of a population. That's sort of a worst case scenario. But some potential benefits is that you also can have refuge populations that have avoided that disease and then can serve through these pelagic larval phase as a way to repopulate affected areas. But also because you're having millions of these larvae, you have a high adaptive potential to sort of adapt to these diseases, and those survivors can then repopulate and potentially have some resistance. Next. Which leads us to, how do we manage disease, particularly in a wild population? You can imagine it's going to be very difficult to treat wild populations of animals. So what we have to think about is more of a management situation. And this can also be difficult, and we don't have a lot of experience managing diseases in marine populations. And one of the problems for this too is normal for many of these species haven't really been characterized. When we're approaching, as a veterinarian, when we're approaching a disease situation in an animal, we always want to compare that to what is normal. And it can be very difficult if we don't really know what normal is. And there's also there isn't a lot of historical information on diseases. So we're really almost starting from zero. And then you top that off with the fact that we don't really have a central database for reporting disease observations. It makes it very difficult to get going to characterize these issues. So when we do think about management, typically, that is the only approach. We can't treat. So our management approaches for wild populations are protection, um, creating refuges where you can't fish, you can't collect. We may limit harvest and transportation or movement on those animal species, just trying to keep a disease outbreak localized in a particular area. If it's a severe situation where we've lost a lot of a particular species, where we're worried about extinction or extirpation, then we might look at captive propagation, bringing some of those animals into captivity and trying to propagate them in an aquarium or in a research setting with the idea of potentially being able to repopulate. And those are really the tools we have in the toolbox when we're thinking about marine disease events. Next, I think we're moving into the next section where we're going to learn a little bit about PRIMED itself.
Thanks, Tim. So this is Sarah, and to build on what Tim was saying about how difficult it is to observe and detect marine diseases and the animals that have them, we are forming the PRIMED network. PRIME network stands for Primary Responders in Marine Emergent Disease. We are primed for the future. And the backstory of this is I'm a sea star wasting disease ecologist. I studied the sea stars before the wasting disease occurred. I was really interested in how sea stars as a predator shape their communities. And suddenly all my research subjects are gone. And as the outbreak came through, we did a lot of things really well as a scientific community, and we also lost a lot of opportunities. So things we did well were tracking how it affected, especially Pysastra ocraceus, the ochre star that we see in the tide pools. We did a pretty good job of tracking how it affected Pycnopodia, the sunflower star. We did a worse job at tracking how it affected the like 10, 12 other species that sea star wasting disease affected. And we also did a really good job at collaborating with the network of people who studied sea star wasting. And it became very apparent as we were forming these collaborations and having meetings and having workshops about sea star wasting that networking was crucial to answering all the questions that we had. But some of the things that we didn't do well were essentially identifying the cause. There were people who made valiant efforts and did a lot of work quickly, but we had a very narrow time window to really sleuth out the disease. And we essentially as a scientific community, did a handful of experiments that pointed to this virus. But when we redid those experiments the next year, that they didn't pan out again. And so now we're back kind of where we don't really know the actual virus or pathogen that caused this disease. And some people aren't even sure if it was a pathogen. But essentially, we've passed the time window where we have animals that aren't exposed. And it's really hard to do an experiment on an animal when none of your animals have never seen the disease before. And so we had a time window that we essentially missed. And it occurred to us as a group that if we had been more preemptively prepared for an outbreak, we may have done a better job at characterizing the wasting disease, both as the cause and how it affected a bunch of other species. So we formed the Sea Star Wasting Disease Task Force back in 2014. And as a member of that task force, I ended up working a bit with some folks from the Association of Fish and Wildlife Agencies. It's a national organization of all the state agencies. And in the works at the time was the Marine Wildlife Health Act, because there is no federal level apparatus that's meant to deal with wildlife diseases on a national scale. It's left to states. And so as I was working with these folks who studied bats and salamanders, they turned to me one day and were like, can't you just go vaccinate the sea stars? And then another was like, can't you isolate some of them and keep them healthy? And others still were like, can't you just make a bunch of babies and put them back out there? And I was just like, no, we can't. Because sea stars have different immune systems. They don't respond to vaccines like mammals do. They are really hard to keep in captivity, and even harder, well, not really hard to keep in captivity, but especially hard to breed in captivity. It takes years because they have that pelagic larval phase that Tim talked about. So it became very apparent that the way we manage diseases in the marine environment, for especially like a marine invertebrate, is very different than how we manage a vertebrate on land, like a bat or a salamander. And so we formed the PRIMED network to specifically address that sort of gap in knowledge and be both better prepared for the next one, and to identify what sort of tools we could lift from the terrestrial world into the marine environment. Next. So the PRIMED network is a group of over 100 scientists and managers and community members. And we have 3 general areas of expertise. The first is diagnostics and disease dynamics. So that's the folks who are able to do the epidemiology and the identify diseases, like someone like Tim, who's a veterinarian, and also understand how diseases transmit. The second group is more up my alley, where I'm really into the population dynamics, how that affects the host population, how the disease might affect other organisms in that trophic cascade, like Silke was talking about, and how the environment might be linked to a disease outbreak. And so for sea star wasting disease, for example, it seems like warmer temperatures make the outbreak worse. And then finally, we have folks who are, their expertise is in mitigation and remediation. So essentially management, trying to either slow diseases or help host populations bounce back after disease. These 3 groups have both a monitoring plan and a responder plan. And so we have folks out there, and we're hoping to help you be part of this monitoring network, with their eyes in the water, their fins in the water, their boots on the tide pools, looking for marine diseases and looking for abnormalities. And then we have another set of plans for what we do when a disease or suspected disease outbreak occurs. The type of people who are involved in PRIMED are people from universities like myself, from state and federal agencies, from non-government organizations, and zoos, aquariums, museums, First Nations entities, and Native American entities, and then finally community scientists, just like you all. And today I really want to focus on that last piece, the community scientists, and how key the group, this group is to PRIMED. And then finally, I just wanted to point out that we're funded by Oregon Sea Grant. We're funded by the Band Foundation, which is based out of DC, and Oregon State University. Next. So the PRIMED mission is to detect, monitor, and effectively respond to marine wildlife disease emergencies in a changing ocean. And some of our goals are to first develop an active network for communication and collaboration. We are well on our way with that one. Our next is to efficiently detect, rapidly respond to, and effectively monitor marine disease emergencies. Next is, during a marine disease emergency, activate our network and study the causes and the consequences, and then also consider the management actions that we could undertake. And then finally, to increase public awareness and opportunities for engagement by local communities, which is why we are here today. Next. So this is a map on our website, primednetwork.org, that shows all of the scientists and researchers and managers that are already part of our network. You can click on any one of these on our webpage and find out a little bit more about the people near you who are part of the Primed group. We are primarily on the US West Coast for now, because we got to start somewhere, and we won't want to go too big too fast. But we do have folks, you know, from Hawaii and Florida who are interested, and we hope that what we are forming now could be easily applied in other regions. Next. And with that, I think, oh, it's up to Lily now. Thanks, Lily.
Hello again. So if you're wondering what you can do to help, joining our new iNaturalist project, Marine Wildlife Health, would be a great way to do that. We know some of you are familiar with iNaturalist, but for those who aren't, it's as easy as taking a picture and submitting it to an app on your phone. All of the observations for marine wildlife health will be used to detect marine disease outbreaks and build baseline data of healthy organisms. Early detection of marine disease outbreaks is crucial for a quick and effective response, and we need your help. So if you're interested, I encourage you to attend the Coast Watch sponsored iNaturalist webinar next Thursday, October 22nd from 11 a.m. to 12 p.m. During the webinar, we'll teach you exactly how to use iNaturalist and submit observations in more detail. If you're unable to attend, there will be a recording posted on YouTube that you can watch later. Next slide, please. So we want pictures of everything: healthy, diseased, unknown health, and even unknown organisms. You have the option to add in disease observations, but if you aren't sure, Primed Network and iNaturalist community members can add those in later. What I like most about using iNaturalist is learning to identify the organisms I encounter in nature. I love taking pictures of like plants and animals that I see, and it's exciting that there's a place that I can go to not only share those pictures with other people who are excited about nature, but also participate in projects like marine wildlife health or tracking sea star wasting, where my pictures become observations, and those observations have the potential to become data for environmental research. Thanks.
Hello, this is William. Oh, I don't have any video, but I don't need video, I guess. Hello, so how will we use your data? All prime tagged observations will show real time on our map. So in short words, that means by using the primed health status tag in your project, or by adding your observations to the Marine Wildlife Health Project, you enable your observations to immediately be eligible for assessment with the Prime Network's diagnostics and disease experts. So each observation on the map is actually clickable and links directly to the observation that shows, in this case, these are disease that you see off to the right. And then you're able to see immediately what is diseased in your area, and maybe you could go out and help search for some in that area also. Search will go out to Prime members, and protocols are currently being developed to create algorithms that will distribute diseased or injured observations for further scrutiny. The health status will be confirmed with specialists through our network. And through our network, experts then can connect directly with observers through the iNaturalist platform, being able to locate under the observation and actually email or contact the person that saw that, so that hopefully we can get real-time information so that there is no lag from when we start seeing things outbreak to when we can get them reported to the appropriate people. And if you'd like to see more about that, our website here is listed at the bottom, and we encourage you to go and check things out. I guess the next person is Laurel.
All right, so that actually concludes what we have for you today. But we have, I think, Jesse, a little bit of time for questions about what we presented today. And we'd also like to leave you with this plug for our upcoming workshop, where we'll, as Lily said, teach you how to submit observations to us, so that we'll see organisms that are both healthy or maybe diseased. So that's all we have today for you. And thanks so much for joining us and having us on. And thank you, Jesse, for arranging this with us. This was so great.
Thank you so much, everyone. That was wonderful. I'm not sure where my video went, but maybe you can see me. That was excellent. So much information. Some of it kind of hard to hear. It's really interesting after kind of following what's been happening for the last 5 or 6 years with sea stars that we're at the beginning because there's so much to learn, of course. And I'm really happy to share this with Coast Watchers. I hope that Coast Watchers become involved. And yes, if there are questions, please start to type them in. We have many people here that would love to answer them. Our first question from John: is there evidence of disease in farmed populations spreading to wild populations, for example, salmon?
I think Tim would be the best to answer that question.
I think there is, although there are a lot of, and again, I haven't worked with salmon for years, so just a minute, I've been asked to start my video. I thought I did. There is some evidence. I don't think it's as strong as you may hear in the media, but certainly it's possible. Anytime you have these animals concentrated in these areas, there's a chance for spread. And I think the bigger concern is some of the releases you probably heard in the media where you had a net pen fail. And that is actually a bigger concern right now, but no, certainly there is a possibility there for that. I think the other thing we worry about with aquaculture is non-native species getting out, and we certainly see that all over the world where species that aren't native to a particular area getting released accidentally, usually. Hopefully that answers your question.
Thank you, Tim. That was great. Let's see, we have a couple of comments. Thank you from Nancy, a great Coast Watch opportunity, wonderful presentations. From Marty, well done all. Thanks for a great presentation and for your valuable work. Question from Jay Brandt, any efforts to raise sunflower stars in captivity?
I'll take that one. Yes, so there is a pilot study essentially going on at University of Washington Friday Harbor Labs, led by Dr. Jason Hoden and funded by the Nature Conservancy. Jason has about 25 adults in his tanks at Friday Harbor, and he's been breeding them for the last year or so. And he has little tiny baby sea stars that are about 6 months old now. I think they're like smaller than your fingernail for sure. And there are some of them at the Seattle Aquarium. Actually, I think they were, and then with COVID, they took them back out to Friday Harbor. But the purpose of what Jason's working on is to essentially figure out the best ways to raise babies quickly and what are the best temperatures, what's the best food, that kind of thing. And then the next phase would be to scale that up and actually start doing, instead of having 25 adults, have hundreds of adults and multiple places growing baby sea stars, with the intent to eventually use them to repopulate a given area. So that's happening. I think it's happening slowly because baby sea stars grow slowly. They start out microscopic as larvae, and they take years to even get, definitely years to get adult size, but like a couple years to even get like the size of a quarter. So it's not going to be a quick solution, but one good thing is they make millions of babies, right? So what we can do is make a lot of them, maybe put them out when they're real small, and hopefully some will survive, rather than trying to wait until they're adults and put them out in 10 years. That would be a little less efficient, right? Yeah. So I hope that answers your question.
Thanks, Sarah. Another question, as sea stars are recovering, is the urchin population decreasing and the kelp recovering? That's a great question. Thanks, Nancy.
Yeah, I think I'll take that one too. So Nancy, thanks for the question. So there's the difference here is there are actually two sea star species. The ochre star is the one you see in the tide pools with 5 arms. And that one is recovering fairly well. So at least in Oregon, we're seeing bounce backs. The sunflower star, and the tide pool one, the ochre star, does not eat sea urchins. They only eat intertidal organisms like mussels and barnacles and stuff. The sea star that eats sea urchins is the sunflower star, and they are not showing signs of recovery. They are effectively, I have actually a paper coming out in about a month, and we are going to show that they are effectively gone in California and Oregon and most of Washington. There are some left in British Columbia, and there are a decent number left in Alaska. But they're not bouncing back regardless of where we're talking about. So even in Alaska, they've gotten hit less hard, but they're not coming back. So I guess your question was about the effects on urchins and kelp. What we're seeing right now is they're not coming back. The urchins are increasing and the kelp is declining. In places where they are still there, we're not seeing that urchin take over as much as we are in places where they're gone. So I hope that gets at your urchin kelp question.
Thank you, Sarah. Another question from Nancy asks, what to do if you find a dead or dying sea star? What do you do? Well,
First, we'd ask that you take a picture of it and put it on iNaturalist through the Marine Wildlife Health. At least for wasting disease, we can visually assess like that looks like wasting, that doesn't look like wasting pretty easily. As far as like taking it in and the actual animal in and bringing it up to a place for examination, I don't think there really is a infrastructure for that. Tim, there's no places that do that, right?
No, I think the preference would be if you would post the picture and leave it in place, and then we can make a decision whether or not it needs to be sampled. I get terrified of potentially 6 sea stars showing up on our door and then trying to work them up. I think it would be easier for us to just have the image and know where they are, and then we can go out. And if sampling is necessary, sample appropriately on site. Because also that by looking at the area around where that animal is, it can also give us a lot of really valuable information as opposed to just having a potentially 6 sea star in my hand.
Yeah, and there are enough 6 sea stars out there that like us getting access to them is not the issue. It's us understanding where it's happening when is what we are needing from folks like you.
Thank you, Sarah and Tim. Does anyone else have any questions? Okay, well, perfect. It is noon. That was wonderful. Thank you so much, everyone. Again, next week at 11 o'clock, same day. A number of you have already registered, so we look forward to seeing you next week. I just want to thank all of you for putting together such a great presentation today. I imagine that Coast Watchers will be participating in this as they are on their miles. I also wanted to remind folks too that back in, gosh, I think it was March maybe, back when all this pandemic started, we did have a webinar with Melissa Miner from Marine, and she talked about sea star observation logs. And that's another way of getting your photos and some of your observations into the pipeline. And I think, Sarah, do you work kind of closely with Marine? Are they a partner of this? Could you talk about that for a minute?
Yeah, Marine is part of the PRIMED network for sure. The Marine, the seastarwasting.org observation log is actually actually the preferred way you log a sea star wasting observation, just because the marine log has like a bunch more fields than the marine wildlife health platform. The marine wildlife health platform is meant to be like a one-stop shop for any dead animal mortality event, weird looking thing that you might encounter on the shore, whereas seastarwasting.org is very specific for sea star wasting, because we know a lot more about that one, right? You can say like, does it have lesions? Does it have arm losses? And that kind of thing. So one thing I would say is, if you are gonna submit data, submit it either place, but don't submit it to both, because then we're double counting your observations.
Okay.
So
Very good.
pick one or the other, and we'll get it either way.
Very, very good to know. And we can talk more about that next week as well. These specific places to put this, and this is of course today is about PRIMED and why the network is so important. And you did such a great job, and I'm looking forward to learning more and also being a part of this myself. So thank you, everybody. And who will be back next week with us? It'll be Laurel and Lily. Sarah, are you here next week too?
Yeah, I'll be there next week.
Okay.
Yeah,
Wonderful.
probably Laurel, Lily, and I. Maybe
Okay.
Bill.
Okay, great. All right, well, thanks everybody for their time. Again, this will be available on our YouTube channel in the next couple of weeks, and I'll send out a message when that is ready. And I also want to remind folks out there, Coast Watchers and others, the King Tides kickoff is starting on October 28th. We'll have a webinar then. Let me know if you would like to come to that, and have a great day, everybody. Thanks again.
Thank you.
Thanks so much
Thank
for having
you.
us.
Thanks, Jesse.
Thank you.
Thanks, everyone.
Bye.
Bye.